My name is Piotr Bednarczyk, and I'm working in the Department of Physics and Biophysics Institute of Biology at the Warsaw University of Life Sciences in Poland. At the first, I would like to thank for organizers for inviting me and giving the possibility to present my results. Today, I would like to focus on the role of the mitochondrial potassium channels in damage caused by urban particles matters. So, my topic will be concentrate on the mitochondria, on the inner mitochondrial membrane potassium channels and particle matters, which cause a lot of diseases at our respiratory systems. In our research, we use the samples for biological repetition of our experiments from the National Institute of the Standards and Technology, with the diameter less than 4 micrometers. So, mitochondria are powerful powerhouses of the cells, and they have some mainly function like production of ATP, storage of calcium ions. Also, mitochondria take part in the processes like apoptosis, regulation of cellular metabolism, regulation of the and signaling through the mitochondria bioreactive oxygen species. Mitochondria are surrounded by two membranes, outer and inner. Inner mitochondrial membrane is very selective for ions. Mitochondria in mitochondrial membranes consist a lot of ways for ions, especially for potassium and anions. Due to very huge polarization of the inner mitochondrial membranes, the flux of the potassium is very useful. Till now, it was reported that expression of the potassium channels are in the brain, heart, liver, skeletal muscle, skin, and also at the last time in our laboratory we identified the mitochondrial BK channels in mitochondria of human bronchial epithelial cells. Potassium transport into the mitochondrial matrix can change mitochondrial membrane potential, affects the mitochondrial matrix swelling, also regulates the level of reactive oxygen species, and also change the transport of calcium into mitochondria. Today, it is well known that potassium channels, especially these expressed in the inner mitochondrial membrane, are connected with the protection. This mechanism of protection is well correlated with the influx of potassium into the matrix mitochondrial. So, after activation of mitochondrial potassium channels, using, for example, diazoxide, NS11021 or quercetin and others activators, it was observed cytoprotective effects of the cells, but detailed mechanism of this is still unknown. So we ask the question, can we protect epithelial cells against damage caused by urban particles matters by activation of the BK channels, mitochondrial BK channels? In our research, we use two cells models. These human bronchial epithelial cells we use as a wild type cells, and after using CRISPR-Cas9 technology for using disruption of the KCNMA1 gene, which encodes the alpha subunits responsible for forming the pore of the BK channels, we use these HBA delta alpha BK channels with deletion of the gene encoding of the BK channels. Also, during the experiments which I would like to show you, I will use some inhibitors of the channels like penitram A. Also, we use the quercetin activators of the BK channels, and for some controls experiments we use isaremnetin, and the differences between the quercetin isaremnetin have some methoxy group in the third prime position of the B ring of the molecules. So, the first we start prepare the patch clamp experiments, And as a first, I tell you that we discovered this channel in the mytoplast from bronchial epithelial cell line. And these traces show the control activity of the channel. The control activity channel have a open probability about 90%. After application, the 10 micromolar concentration of the quercetin, we see increase activation of the channel about 60–70%. After the treatment, quercetin with particulate A, which is the inhibitor of these channels, we observe strongly inhibitory effects after 4 minutes. So we have the quercetin as an activator and particulate A as an inhibitor of the channel. As I tell you before, we use the isomethylene with this group, methoxy group, and in our experience we don't see any changes in the activity of the channels. The open probability do not change after application, after incubation, our patch with the 10 micromolar isomethylene. Using these results, we use the technology for high-resolution respirometry, and we measure oxygen flux. The black bars is bars when we apply the quercetin, different concentration, and after application of quercetin, we see huge changes of the flux, oxygen flux. In the presence of the inhibitor of the channel, the flux was not present. This indicates that channel activation can change the respiratory rate in our whole HbA cell models. As you see here, the particulate A with isomethylene and isomethylene alone cannot change the activity oxygen flux in our Y-type HbA models. Also, we measure membrane potential, and after application, different concentration of the quercetin, we see the polarization of the membrane. This means that opening of the channels can depolarize the mitochondria. This effect was not observed in the presence of the isomethylene. Also, we measure oxygen level in the presence of the particulate matters. So, what was surprise that we observed after application, after incubation, during the 24-hour with our particle matters, we see very huge changes of the basal respiration of the cells. Also, when we use some uncoupling system, with using the FCCP, this change of the oxygen flux was very huge. But when we play with the quercetin and particle matters, we do not observe, we do not reverse this effect with using the activator like quercetin. Similar effect was observed when we measure respiration oxygen flux with using these cells with deletion of the alpha BK channels. This means when channel is not present in our cell models, we also see a very low oxygen flux in this type of the cells. As a last set of the experiments, I would like to show you experiments with transepithelial electrical resistance measurements, TEER. We collect the cells in the bottle, then we transfer the cells to the inserts, and after getting about the 300 ohms multiply square centimeter, we can start the measure in our system. Some cartoon how we prepare this type of the experiments is here. 0, it's a time when we start the measurements and application of the particle method was started at 0 time. Before 1 hour we play with the substances isomethane and quercetin, and in this last point we use the penitram A as a first, as an inhibitor of the channels. After we add substances like quercetin and isomethane. So what we get? We get information. Maybe we start from this solid line. This line represents control measurements, and after incubation cells with particles matters, we see huge decrease of the transepithelial resistance, disruption of the layer of the cells. But when we play with the quercetin with particle matters, we observe strongly increase of the resistance of the cell layer. The penitram 8 a little changed this effect through the inhibitory effects of the channel. So please see here for the minutes. Also we have a control line and application of the particle matters decrease the resistance of the layer of the cell layer, like in this situation, but application of isomethane and isomethane with penitram do not change the our transmembrane epithelial cell resistance. Similar effects we observed when we compare our experiments with this line HBA with deletion of the alpha BK channels. These experiments indicate that we can create the barrier with using this cell, but we from the sometime we lost the integrity of the epithelial cells in the cells without channel. At the end, I would like to show you some experiments with using the trip and blue methodology. We measure the cell viability, and after application of the particle matters with concentration of 50 micrograms per milliliters, we observe decrease the cell viability. And also this cell viability is observed when was applied when system was incubated with isomethane. Quercetin reversed this effect, and probably this is the effect of the activation of the channels. At the end, I would like to summarize my talk. We can say that BK channel present in the inner mitochondrial membrane also is important player for the bronchial epithelial cell physiology. Activation of the mito-BK channel can be involved in cell production against particle matters induced damage. And at the end, I would like to thank for my members of the lab, Kamila, Agnieszka, Mirek, and PhD students Ada and Jakub. Also, I would like to thank for Bogusz and Karolina from Adam Szewczyk Labs. My study was supported by National Science Center from Poland. Thank you very much.